I just wanted to tell you about the submission of your preliminary descriptions. I have slowly started to give, I think I've only sent about 3 of you feedback, so it's feedback on what you sent me, and I'm suggesting 2 to 3 different articles. If you have received that, I'm just verifying this in all of the files I'm sending you. These are suggestions. I'm not saying that you have to include them in your presentation, whether it's capsule or final presentation. It's just some suggestions in case you're probably doing more research about this topic than me. All I did was spend about 20 minutes on each one of you searching for articles and figuring out what might help. But don't feel like you have to talk about it or incorporate these topics in your presentations. Okay, so now we're going to start talking with no more photosynthesis. We're going to start talking about more plant overall. So by the first line, we start. We've been talking a lot of interactions of plants with their environment, how plants adapt to their environment. So how they respond to light and to pathogens and to herbivores, and all of that focus on how water moves, on how stomach and all photosynthesis, but clearly, just like us, we have all molecules that help us regulate and communicate with some cells, basically what the rest of the organism should do. And this is not limited to plants. Like, how can certain signal that's received in one cell affect the characteristics of this cell? Do humans do that? Might be overthinking. Yes.
I can't go into much detail, but through like signal pathways.
Yeah, so signaling pathways will tell one cell what to do, and then you have different ways in which you can reach the same cell or the cell next door or the cell far away. What do you call that? The ones that go and reach cells far away. Yes, but
Endocrine signal.
yeah, so basically if you have hormones that are telling your body what to do. So you have insulin, and then in response to insulin, these stem cells will learn how to do. This is very important in plants because plants have no cell migration. So there is really no way for one cell somewhere to navigate and tell a different cell something else that they should be doing. And probably you can start relating to that with this example. You probably have plants at home or have seen them in. What happens when you have a plant and then you remove these terminal bud in the plant when you pinch it? Yes.
Any signal to your gut?
But think at home. So let's stop thinking signal. You're at home and you pinch your plant. What happens to the plant? Yes.
It stops growing more vertically and more vertically.
Yeah. So it branches more. So whenever you have a plant and it's growing too tall, and then you pinch and you remove this part, it starts growing. It starts getting all bushes, right? So that is, the pruning or the pinching that you do, and that is apical bud decapitation, because you remove the apical bud, and it's called decapitation, and so you're making your plant just grow more to the sides. Do you know which hormone is responsible for this? It's the most important plant hormone. Well, I don't think it's the most important. It's the one that's important in everything.
Is cytokine?
No, but it's also important. Yes. Yes. So oxy. Okay. So auxin is basically responsible for maintaining this that is called apical dominance. As we're going to be discussing today, it's not just auxin, so it's not going to be clear cut, but we're going to talk about the most important aspects of the hormone. And so whenever what happens is auxin mostly accumulates here in the shoot apical meristem, and it maintains all of the buds silent. Once you remove that, it's like the signal to stay under control disappears, and everybody starts going towards the sense. So how would you define a hormone? Yes.
Signaling molecules that are transported between cells generally.
Yeah, so a signaling molecule, right? So it's a chemical that can affect cellular processes in distant cells. And one of the key things is that you only need a low concentration. You don't need a whole lot of that to cause the distant cells to act or to adapt to that. So we have 7 hormones. And I am going to just take a wild guess, and it's probably the first time you're going to be learning anything about them. So we can tell you today's lecture and the next lecture are probably going to be dense because it's a lot of new material coming in. So, and plus on top of that, they all interact. So it's never just one single thing. But so what I try to do is kind of give you some examples, try to find plants. The way scientists many times figure out what something does is they delete it or they overexpress it and then they see what happens. So maybe in that way you can put more like 2 and 2 together and figure out the important role or the most important roles of each hormone. So a couple of things I am going to focus on the hormone name, the name of the active form, the precursor, and what I'm going to tell you about precursors is not so much that you have to remember the name of the precursors, but sometimes many of them, the precursors are going to be products of secondary metabolism, and because we're going to start talking about secondary metabolism soon, so at least you start hearing some of these names of these compounds and the major effects that are going. So let's start with oxygen. Can anybody tell me anything about oxygen? Yes.
I think if I remember correctly, it's a growth enhancer. Helps speed up growth for the plant. I know it's pretty heavily regulated because of it. Like it's unable to induce growth. So I don't know too much other random information.
Yeah. So growth enhancer induces growth. Heavily regulated because, well, we're going to see what happens when plants grow too much. Auxin is everywhere. I mean, I don't think there's, at some point, you are going to get to the least important hormone, and I'm going to say, but auxin can still override. So, just take you, auxin is 30% the answer is auxin. 30% of the time. So the active form is known as linoleic acid or IAA. This is, again, if you in any of the things, the papers that you're reading, you read IAA, that's what it is. They are not explaining you, we are trying to stimulate auxin. No, they are saying we applied IAA and they are assuming that you know, because it's like telling you, we explaining someone why they are using insulin. So it's like, down the world, that's what it means. Everybody expects you to know that. And it's from tryptophan, so clearly you know from an amino acid, you can have your own.
Well, since the YUC is going to help you with conversion from the TRP to the IAA, if you have an overexpression of it, then any well, maybe not any, but maybe most of the TRP that a plant would have is getting converted to IA. So they would be having an overexpression of auxin.
Yeah, so they are going to have a lot of auxin. Why can you think of a phenotype? So that's good. So you see, this is I said before that this is the precursor. So if you're overexpressing this, you have more oxygen. If you're overexpressing it now, the plant has lost the ability to regulate it. You're just reducing oxygen massively. Based on what you know of oxygen, how would you expect a plant that is overproducing oxygen to look like? Yes.
I mean, it would just have reduced growth and it could have excess nitrogen based on the
Why reduce growth?
because you said it's a growth enhancer, it would have increased growth.
Yeah. So when you think of a plant increased growth, what does that mean?
It could be, I mean, you said it impacts most of the plant, so it could be taller, it could be larger leaves, it could be larger branches.
Yeah, so it's going to be overall, taller plant. And so this would be the wild type plant. This is an Arabidopsis plant. And then the plant that is overexpressing that gene is overall taller. So in the internodes, everything is just normal. So the internodes basically are lower. So essential for growth plays a role in virtually everything you can think of plant. Promotes cell elongation. So cells just become longer. It doesn't necessarily mean they are dividing, they just elongate. They grow. An important thing also is delay senescence. So keeps plants looking young and green. But pretty much, as you're going to see, it does take effect. Yeah.
Okay, I just had a question about that. So because the plants, they're taller, but if you give them the same nutrients, would they not be weaker because they're more extended than, say, a shorter, denser
Yes.
plant?
So yeah, well, so in this case, I think eventually, so one of the most widely used herbicides, actually in chili plants, stimulates auxin production in plants, and the plants, what they do is they grow, and they grow so fast that they exhaust the nutrients in the soil and they die. So they have no control. You're forcing them to grow. They grow, and they, of course, start stealing nutrients of other competitors and other weeds, and then they grow a lot and they die. So yes, plant can. This is a small pod. Arabidopsis doesn't survive for a long time, but I'm assuming eventually, yes, if it overgrows, it's going to be. So it doesn't necessarily mean it's good news for them. And as I briefly mentioned at the beginning, one of the most important things is apical dominance. Apical dominance when the plant is growing more taller than in size. This is very, very important when we talk about plant domestication. So whenever people have selected, or people over thousands of years have bred for better plants, sometimes you're going to see, depending on the plant, you want to select apical dominance. For example, when you think of a corn plant, you don't want so many internodes, right? You want more like stronger main stem than less bushy thing. So this is a very important factor when plant selection and plant breeding. The exact mechanism is unknown, but what is known is that the terminal bud here has a lot of auxin, and then it spreads it downwards. And if there's plenty of auxin in these basically lateral buds, then that stops their growth. If you decapitate the plant, that signal goes away, so now the lateral buds can grow. And so just to not get into any more details, auxin is important in phototropism, so growth towards the light. You've seen that plants, if you give it light to some side, it might grow towards that side more. It's important for geotropism, for the growth of the roots, and also hydrotropism. So apparently there are some sensors that can detect to some extent water potential changes in the soil, and then there you put solution to it. So guides plants grow in the direction inside the person. That's all. We could talk about oxygen.
Pros.
Yes.
What converts it from like the inactive to the active form?
Yeah, so in this case, this gene is going to be regulated by so many other things. So this is the active form. The active form is the endolacetic acid, and this is the precursor.
Okay.
It's not like a one direct step, but this is regulated like, like whenever you are releasing or producing insulin, or releasing or producing hormones.
Okay.
There are many different environmental factors that are going to affect certain gene expression.
You showed like the initial form was like indole 3 acetic acid. Is that like a stored form of it?
No, so indolacetic acid is the active form.
Oh, okay. I was looking at because the structure is slightly different. But
no,
it's just drawn differently.
Yeah.
Yeah. Okay. Cool.
It's just
that
I
makes
should have put the same one, but yeah.
Sweet.
I think this is the one maybe the book has probably. That's what put it on.
Okay. Cool.
Okay. The second hormone is gibberellin. Can you tell me anything at all about gibberellin? Yes.
I'm not 100% sure, but I think because there's several different variations of it, but I think a lot of it has to do with cell cycle communication.
Like,
I mean, I don't know all of the details. I want to say it was like it's needed for transitioning between like cell development as far as like organelles and stuff like that. But I'm not really.
something with cell division. Yes, but I think that's the landmark of cytokines.
That's why I didn't want to say division because I mean that's.
Yeah, okay, maybe cell cycle. Yeah.
It's an electron donor for cell cycle, would that be what it does?
It's an electron donor for the cell cycle.
Or sorry, just based off what's up there, it has the R's can either be oxidized or reduced, and it looks like it has the two forms, so it could have, that could be some limitation.
I don't, that part I don't know. Could be, I cannot tell you yes or no. Maybe I'll have to check that out. But these are different forms. So gibberellin have different multiple active forms. So it can be GA3 to GA7. These are just different active forms. What about it's a hormone clearly, so what does it regulate? What does it promote? What does it relate? What does it do? How was it discovered? Anything at all? Yeah.
Isn't it involved in like termination a little bit? I don't really remember much more.
Termination, promoting, or delay?
I'm gonna say promoted. Delay.
Well, it has, termination is pretty important, so we're going to talk about that in a minute. We'll figure it out. So, many different forms. So, if you just say, you're going to probably see that gibberellic acid or GA. Whenever you see GA, that's going to be gibberellin. Many forms. The reason why I put this here is that the inactive forms, the precursors, are terpenes, which is a very important type of secondary metabolite. It's a type, like a family of secondary metabolites. And so you can have some of those secondary metabolites converted into gibberellin. So you have the precursor and you have a gene that converts the precursor into the active form. You delete it. Which one is more likely to be represented with wild type and which one is the think about what gibberellin does, and then if you delete it, would you expect it to be taller or would you expect it to be shorter? Yes.
Would be shorter because it promotes cell elongation.
Yes. So it would be shorter because this one more than yeah. So overall plant growth, I guess, cell elongation. So it would be shorter. So in this case, when we have this would be the wild type, and this would be the new. So you have a dwarf phenotype. If you delete, if you impair gibberellic acid, then the plants are going to be dwarf. So it's involved in cell elongation, seed germination, flowering, pollen, everywhere. We're still talking about. I believe the three first hormones, auxin, gibberellic acid, and cytokinin, are like all doing everything. But so seed germination.
Well, I was just thinking, doesn't jasmonic acid also do pollen development as well?
Yeah, so jasmonic acid, I mean, auxin also does cell elongation.
Yeah.
So,
Like, how would you identify like the difference? Because like they look really similar between auxin versus.
yeah, so one of the things is, for example, like, if I just told you, this is the wild type, this is the mutant, what's wrong with this plant? That's not a fair question, because it could be auxin, it could be cytokinin, it could be almost everything can be, unless, basically. So you cannot really have one specific phenotype. So what happens is, all of these experiments are, you delete it and then you see the effect. You induce it and then you see the effect. Or many times what happens is you can actually spray the plants with the hormone, and the plants will, like, if you spray a plant with JA, it's going to think JA is perceived. That's what we talk about chromatin in the pathogen. You can spray JA, and the plant is going to induce JA related pathways and suppress SA and become susceptible to pathogens. So that's how they know. And then in this case, we're only looking at this. There is an effect in the roots, there is going to be an effect in flowering, there's going to be an effect in fruit development. So I think for the most part, what scientists do is they all somewhat contribute a little bit, and they isolate which ones contribute the most and where the balance is. But I don't think you can really just isolate it as a single one. One of the interesting things about gibberellins is fruit size. So what happened is, grapes have seeds. I'm saying that because most grapes now don't have a bunch of seeds, but before all grapes had a lot of seeds. And so what happened is someone at some point discovered that you could produce grape plants that were seedless. The problem was the seedless grapes. The grapes themselves were smaller. And people couldn't figure out why. I mean, the seeds are just supposed to be there. Why are the little seeds inside related whatsoever with the actual size of the grapes? And if you're a grape grower who's trying to sell these, yeah, it's great, I have no seeds, that's appealing, but I don't want the grapes themselves to be tiny. So they very quickly found that seeds are actually a source of a growth regulator. Later on, they figured out it's gibberellic acid. So to some extent, many seeds also control or release gibberellic acid, and that causes the fruit itself to grow well. So scientists were in this kind of, well, what do? We want seedless grapes, but we want grapes to be longer. So they found that developing seeds produce gibberellins, and what happens is the gas produced by the seeds diffuses into mature tissues and promotes growth. So this guy is holding seedless grapes and then here seedless grapes, but obtain finally a good size. How can we obtain large seedless grapes? We have them.
Just like spray them with
Yeah.
normal.
You spray them with gibberellic acid. So there's no need to mutate anything, it's just you spray them with gibberellic acid. Now that said, you can still breed and select certain varieties that are seedless but have larger grapes. So it's not that you just have to spray. But depending on the variety, then you spray gibberellic acid. How many times? It depends also on the grapes. It can be 1 time, it can be 3 times, it can be 4 times over the season. And then you just have seedless grapes but larger. So gibberellin stimulates stem elongation and flowering. So here is these regular cabbage plants, and then these ones that have been sprayed with gibberellic acid. So massive.
I guess would be like number 2 would be susceptible because it's like it kind of grew a little bit more like unregulated than the water version. So like assuming that the bacteria would be kind of like producing some gibberellin, so it's susceptible and be higher gibberellin expression.
Yeah, so actually, Sam, you mentioned something important. So in this case, so Gibberella fungicori is, well, I don't want to get into fungal biology, but it's the sexual version of Fusarium. Fungi can have sexual and asexual reproductions, and they have different names, and they look different, and they are like 2 different beings. So that said, Fusarium is a very important plant pathogen. Call it different species, and you have some of the most important plant pathogens. And what happened was actually gibberellins were discovered because some plants in the field were infected with Gibberella fungicori, and the plants were taller. They were just significantly taller. So it doesn't make sense that if you're infected with a pathogen, you're going to be taller, because we usually associate taller with healthier and shorter with sick. So what happens is scientists soon, well, I don't know how soon, but eventually they realized that Gibberella, this pathogen, induce or actually produce gibberellic acid. And it's like exogenously applying gibberellic acid. It causes the plants to be elongated. And this is the picture the beginning has. So Gibberella produces gibberellic acid. Plants grow very tall, and so one of the things we call they lodge. They become weak, and then they eventually lodge. Why do you think a pathogen, how would it benefit a pathogen to make a plant grow taller? You're a pathogen and it's like, here's an idea, I'm going to make this plant grow really tall. Why would a pathogen want to do that?
It would be able to have more photosynthesis, so you get more nutrients, and then once you kill it off, it would be stronger, or you could get more nutrients.
So why do you think more photosynthesis? Because there's more
It just allows for more growth and better competition against those in the area.
Okay, so I'm just trying to figure out how do you think most of this is because there's more leaf area or?
Correct.
Okay. Yeah, I mean, but wouldn't they accomplish that even if you grow to the size?
Yes.
So, I mean, but yeah, I mean, if you make it grow more and then there's more tissue, you're already in. You probably want it to grow more and then, yeah, you have more food. Yes.
Well, I was thinking probably because the plant would be so focused on its growth because of the hormone, it'd be easier, or it would be making a weaker target because it'd be so focused on its producing things for growth, which then also, whatever the pathogen is, could also intercept those and use those for their own growth. But by the plant being so focused on growth, it's not going to be so worried about the pathogen. The pathogen can go a little bit more undetected to its different pathways.
Okay, so that's a possibility, and I think that's one of the things is when the plant is focused on growing, and when you're sick, whenever you feel sick, your body actually is like stop, focus on spending your energy on fighting this disease that you have. Go to bed, stay in bed, and recover. This is not allowing the plant maybe to allocate some of the resources and it's focusing them on growth. It's quite tolerant.
The plant is going to lodge, does that mean if it like, obviously falls like to the ground, basically, would it then be easier to access to like decompose for the fungi?
Yeah, but the fungus is already in the plant.
I didn't think about this until now. But if it's a fungus, the height would help it because it's going to release spores. If it can get up higher, it's going to have more access to release spores downward.
Yeah.
So easier to spread.
So actually, everything that you said. So tell you, scientists still don't know. I mean, because how do you say it's just for this but not for that? You're not the plant and you're not the fungus. So you might be overlooking something. However, all the experiments suggest that the reason for growing taller is the spore release. You have an advantage if you can spore release and then fly away. Why do they think this? Because it's an airborne, well not airborne, but these the spores of these pathogens are usually spread yes by water, but also by air. Also, when you think water, if you're taller and it rains over here, you're easily splashing into other plants without you doing much work. But a lot of ideas there. But yeah, they don't know why it is. Okay, let's start talking about cytokines. Another form there. Active form, we have multiple active forms. And let's just call it cytokines. And it's from iron. It's how it is. What can you tell me? Anything you can tell me about cytokines? What can you tell me about cytokines? Anything whatsoever that you know.
Does it have to do with cell division?
Yes, it's important in cell division. And if I had to label, the most important thing of cytokine would probably be cell division. Cytokinesis, cytokine. That's actually what it was called, cytokine. So I guess I give you the question, the answer. These are base. So these are all the leaves. So you have the wild type here and 2 mutants. I'm telling you, ROC2 and ROC3 mutants have defects in cytokine metabolism. Compared to the wild type, are they decreasing or inducing synthesis of cytokine? These mutants. What?
Or cytokinin also did millions of senescence, so if it is surviving for longer, then it has more cytokine.
Yes. So cytokine also delays senescence. So one of the where I like to associate them is if you promote cell division, when things stop dividing, eventually start senescing. But if you are constantly dividing your young because you have newer cells are dividing. So cytokinin also, in this case, they are inducing synthesis of cytokine because you can see the wild type is completely senesced and dead by day 28. However, these mutants can go longer periods being green, so it's delaying senescence. So cytokines are also important in inducing zinc effects. So we've talked about the zinc effect in which the fully mature leaves are the sources that provide photosynthesis. And the younger leaves would be here are zincs. Until they are not fully developed, all they are doing is being zincs. Remember that we talked about how elements that can be mobilized through the phloem, the plant will prefer to give them to the young leaves that are still being zincs and are developing. So for a period of time, only fully developed mature leaves are sources and they provide photosynthesis products to the rest of the plants. So scientists also found that, let's say these are two fully mature leaves, and if you spray cytokine into one of these leaves becomes a zinc. Even if you are a source, they stop being a source and they suddenly become a zinc. Also, if you have a leaf and you spray cytokine into only half of the leaf, that half will not senesce and become a zinc. A zinc. So you think about it, if you become a zinc and you are actually getting photosynthesis from the plant, you're not senescing, you're remaining green because all of the nutrients are coming to you. So this is so evident that there are sometimes some pathogens, if you're ever walking around and you see a leaf that is almost senescing, and then it's almost all yellow and having green spots. These are called green islands. And many pathogens and many insects, actually they can do this. They keep them as zincs, so the plant keeps giving them nutrients, and the pathogen survives in that little spot. So it can be sources of zincs. As long as cytokine is applied or is present in that tissue, it is going to become zinc. So they induce cell division, induce the passage from systemic. That's what I thought we were talking about specifically. So they delay senescence, zincs. Also important for apical dominance to see here is where we started already stepping over auxin. And also it's important for when buds are dormant, they will stop being dormant. So how do they regulate that? When you have more auxin, you have more apical dominance. When you have more cytokinin, you end bud dormancy and then the lateral buds start growing. That's how you have now some kind of regulation. So again, auxin will most of the time overcome the activity of cytokinin as long as you have enough auxin in your shoe. If you remove that, then or the balance eventually, if the buds need to grow, then cytokinin is not so good. Does anyone know anything about cytokinin? Where it's from? Very specific, but did you?
I don't know if it's the answer, but I remember I think isn't there that one bacteria that can cause I'm forgetting its name now. It can cause essentially almost like a cancerous growth because it can increase cell division and it essentially pulls up into a tumor.
Yes, so actually cytokines, because they induce cell division, they are very important in a plant disease that is causing tumors, basically. So they were discovered as a factor promoting cell division in tomato plants. One more thing, what does this tell you about the role of oxygen? Sorry, oxygen. Cytokines, what already cytokines? How is this panel different from this one? This is a mass of cells, undifferentiated plant cells. But you didn't really have to know that for now. But this is a mass of undifferentiated plant cells. And this is also that, but this is only auxin, and this is cytokine. How are they different? Yeah. Yes. So they induce. So think about this. Auxin induces cell growth, but cytokine induces cell division. When you have cell differentiation, you need cell division because if you grow but you don't divide, there's no really cell differentiation. So they induce not only cell division but also cell differentiation and in this case onset of photosynthesis. Right? So they were discovered as a factor promoting cell division in tomato plants. And discovered because it's one of the most important factors involved in these plant disease. So agrobacterial tumefacients is one of the plant disease. That's the least important factor in this plant. It's really not a very important plant. But this is the most efficient method that currently exists to transform plant cells. So if you want to transform a plant cell, this is what you're going to do. No CRISPR, none of that, eventually, but CRISPR hasn't been really well figured out fully for plant cells. So it's this plant pathogen. So why is that? Agrobacterium is a bacterium that infects plants and produces these kind of things. So what happens is this bacteria has something that is not unusual for bacteria, and it's a plasmid. So we know many bacteria have their own genomic DNA, and they have this extra chromosomal part that is a plasmid. Remember, plasmids, in bacteria, plasmids usually carry things that give them an advantage, right? Antibiotic resistance genes, things that are not necessarily required for survival. For agrobacterium, this is called a T plasmid. And the T plasmid carries a bunch of genes that are important for pathogen growth. And what happens is this gets inserted into the plant chromosome. So the bacteria infects the plant. And this red part doesn't just stay floating around, it's randomly inserted in the plant gene, in the plant chromosomes. And some of these genes are cytokinin and auxin producing genes. So now it makes sense. You are forcing the plant to divide and to grow and divide. It forms this. And only you, the pathogen, can grow here. They just force the plant to create other things, not just these hormones, but they create an environment in which, in that tumor, only that pathogen grows. And because it's so far the only known plant pathogen that delivers plasmid into the bacteria, then what scientists do is they remove this red part, and whatever you want to transform that bacteria with, I want to make that plant, I want to genetically engineer that plant and introduce genes that do insert trait. you put it here, and then that's inserted in that, and there it's good. The bacteria is very efficient at penetrating and transforming the plant, so you just have to inoculate your plant with the bacteria and let them do the rest. And so that's what produces the two. There are other things there, like some amino acids and some compounds that don't exist except in that plasmid, except to allow other bacteria. Okay, we have time to talk about ethylene. So ethylene is ethylene. It doesn't really have any major complex structures. It's a gas. It comes from the methionine. So the precursor is methionine. This is the wild type. And these are your mutants. They were all sprayed with ethylene. So for reference, this is how the wild type looks like. One of the mutants is more susceptible to ethylene than normal. The other one is less susceptible to ethylene than normal.
Doesn't ethylene play a role in senescence? So I think if mutant one would be more susceptible to ethylene. It's going into senescence.
Yeah, so ethylene is important in inducing senescence, right? So we talked about how cytokinin delays senescence. Ethylene induces senescence. So it induces chlorophyll degradation, so that's correct. Mutant 1 is more susceptible to ethylene. Mutant 2 doesn't care if ethylene is present, it's staying good.
Well,
One will have the lowest levels of ethylene 1.
you said it induces senescence, so that's the youngest version of fruit. So
Yeah.
I'm gonna go with that.
So one will have the lowest level of ethylene, and it is because ethylene is senescence but also chlorophyll degradation. So that's why. So more than senescence, it starts degrading chlorophyll, so starts senescence. So exogenous ethylene is actually what is most commonly used to induce the ripening of fruits, and it's very commonly used in bananas. So when you transport bananas from 3 or 4 parts of the world that produce the most of them, you transport them green and actually have ethylene captures. So whatever ethylene is produced is like, no, don't pay attention, you have to stay with it. And then, depending on the variety, that most of us just eat Cavendish bananas, everybody has it figured out. So you know X amount of days before you arrive in port and X amount of days before you deliver, you start spraying ethylene and it's already sorted out. So you transport them fully green, prevent ethylene from being released or perceived. And then here you can see, so these A, B, and C, so this is before ethylene spray, 48 hours after and 8 days after. But then also from left to right, you have increased amounts of ethylene that have been sprayed into the fruit. And then you can see there's very heavy. The more ethylene that they have, more mature they will become. Actually, some refrigerators, I don't really know how it works, but my newest refrigerator has an ethylene capsule thing. I don't even know how it works. But they have that. It's a polish that they change. I guess it keeps things from running and just the way you have it. Also, if you ever have For example, I don't like bananas, but I usually have one because that helps me write anything that I want. So if I can go and I buy like green hard avocados, I put it in a bag with a banana, and the next day you have beautiful avocados. Or if you have one avocado that is ready and the other ones are not, put them in a bag, seal it. The next day, almost not fully ready, but it happens. Everybody gets to that. So I think. So it's a plant growth regulator, induces leaf and flower senescence, chlorophyll degradation, and many things. So again, ethylene for me would be ripening and senescence, basically because of ripening. So now let's say you want to do roast. You don't want your tomatoes. The one we talked about, could you spray, counteract, and keep them greenish?
Cytokines.
Sorry?
Cytokines.
Cytokines, that's one option. Other options. Specifically, oxygen. I told you. If you don't know in the exam, oxygen is probably your answer. I mean, you can't be wrong. It's gonna get my grading harder because I'll probably find an article that says oxygen has something to do with it, but it probably does. So it also, so oxygen informing so many things. So when you think about the tomato going from green to red, it's not just chlorophyll degrading. It's chlorophyll, less chlorophyll. Also, the red is many carotenoids are being developed. And also some of the starch is being broken down into soluble sugars. So now you have soluble sugars, this is probably gonna be on the bitter end. This one is gonna be sweeter because some of the starch has become soluble sugars that you can actually taste as sugar. And so what happens is, this is just a very long chart to show you that in the case of tomatoes, you also have carotenoid biosynthesis. And so, as you can see here, it's just a very complex pathway. Oxygen affects some, ethylene affects the other. So that ratio of ethylene to oxygen plays an important role in regulations of many different foods. Okay, so we'll leave it there. We'll start with oxalic acid that I know you're using some liquids as well.